So welcome back to today's lecture. We have treated in our last lecture glycogenesis, how your body can take food and store it as glycogen. So glucose is converted to glycogen by glycogenesis. But in this video, do you know what we are doing? We want to now check. Let us say that we have studied finish, and over a long time we have not eaten. How will the body now convert this stuff to glucose and use it to generate ATP for us? So that's why we are doing glycogenolysis. Glycogenolysis, which means lysis means breaks down of glycogen. Glycogenio, that is breakdown of glycogen. So we start with the introduction. So what is glycogenolysis? It's a metabolic process which involves the breakdown of the stored glycogen, or converting the stored glycogen into glucose used for other biochemical activities. Again, glycogenolysis. Lysis means breakdown. Glycogen means that which is the glycogenolysis means lysis, breakdown of glycogen for other biochemical activity like formation of glucose and use it for ATP. You get it now? So very simple. So now, since we have known this part, so where is all this thing happening? Where is it happening? The first one is it occurs mainly in the liver. All this process occurs mainly in the liver. So the liver is a hallmark where various metabolism occur. Various metabolism occur here. That is why it has the highest cardiac output supply. So the cardiac output to this liver is about 30%. So it's the highest in the body. After the liver, you have the kidneys. So the kidney is the second highest that receive blood more, because they are very important. Remember. So it occurs in the liver. So when you break it down to glucose now undergo what we call glycolysis. So first of all, you have to break it down in the liver. So when you break it down in the liver, they are transported to the muscles and the adipocytes, where they undergo glycolysis to what we call GLUT4. So but every organs have their own glucose transporters. But since we are focusing on the muscle, we are using GLUT4. Okay, now. So now, what is the stimulating and inhibiting factor? You can see it on your screen. Now we switch it. Unlike glycogenesis, formation. Unlike glycogenesis, what is this one used for? This one is just doing when you're fasting. So when you're fasting or you are starving, that is when your body now start, okay, I've not eaten anything for a long time, I have to start breaking down the glycogen I already have. So your body will start breaking down during fasting, or when you are starving, or when you are performing a strenuous exercise. Your body will break down this product, which is glycogen. Also, when your body is now breaking down this product, the hormones that synthesized at this particular point is cortisol, glucagon, catecholamines. All of them, they are all synthesized during fasting, starvation, strenuous exercise. They are usually working. Glucagon, cortisol, catecholamine, they all work at this particular place. You can see it on your screen. This is the only hormones that fuels the breakdown of stored components, so that you can convert them to energy. So which condition we inhibit this pathway? You have what we call fed state. When you eat, it favors glycogenesis, not glycogenolysis. 2, when there's presence of insulin. Glycogenolysis do not like insulin, so when there's insulin, it will not even work. The last one is tyrosine. Remember, tyrosine increases the basal metabolic rate. So, but here you don't have food to increase the basal metabolic rate, so it's not working. So it inhibits it. So since we have said this one, now let us go to the biochemical pathway proper. I get it now. We'll go back to the biochemical pathway proper. So the first one we are using here is what we call formation of limit dextrin. You get a formation of limit dextrin. So this is our step 1. So in formation of limit dextrin, what happened? Remember from that place I said we have 14 glucose units joined by alpha 1 for glycosidic bond. But when we reach the 14th carbon, we see it's too long. The body will now start adding it to alpha 1 6. That means below any glucose molecule, just below it, the body will continue adding it there, forming an alpha 1 6 glycosidic bond. But in this way, if you want to destroy this glycogen, what do you have to do? The body has to do what? Now your body will cut from about, because let's say we arrange about 10 glucose units. So we have 14 glucose units on the horizontal axis, and we have about 10 glucose units on the vertical axis attached by alpha 1 6. The body will count from the beginning. The body will count up to 1 2 3 4. The body will count 4 and cut away. The body will count 4 and what? Cut away. You get it now? So if the body cut 4 glucose units that is attached by alpha 1 6, you are forming what we call a limit dextrin. So a limit dextrin is formed when your body reduce the amount of branched glucose units to about minimum of 4 glucose units attached by what? Alpha 1 6. So 1 4 on the horizontal space, 1 6 on the vertical. But the 1 6 on the vertical, they are all joined to themselves at the first and the 6th carbon. Are you getting now? But if you have 14 on the horizontal, 14 on the vertical, the body will have to cut it until it gets to 4 glucose units attached. You are seeing it on your screen. Until it reaches 4 glucose units attached. By the enzyme, we have what we call glycogen phosphorylase. Are you getting now? Glycogen what? Phosphorylase. So when the body breaks this down now, what will the body do? If you break, you remove it, your body will add it to the non-reducing end of that 14. So we have 14 on the horizontal and we have, let me say, 10 or 14 on the vertical. So if your body counts 4 glucose units from the beginning to the 4th glucose unit, if you cut it at number 5th now, it will transfer them to the other side. Are you getting me? You transfer them to the 14 in the horizontal axis. So that transfer is what we call glycan transferase. So first one, the rate limiting enzyme that allows the cutting, the initial cutting, is what we call glycogen phosphorylase. So glycogen phosphorylase allows the first cutting. So when glycogen phosphorylase allows the first cutting, the next person that will come is to transfer it to the non-reducing end called the glycan transferase. So glycan transferase transfer those cuts from number 5 to, let me say, number 10 or number 14. They transfer it to the non-reducing end of that horizontal 14 glucose unit, joining them by alpha 1 4 glycosidic bond. So let me say, if we have 10 and I go to number 5, I cut. So we have 4 on the horizontal, on the vertical, and we have 6 that we have cut away. So we'll add 6 to that 14, making 20. So on the horizontal now, but joined by alpha 1 4, we have 20 now, and we have 4 hanging. Before we have 14 and we have 10. So if we now cut at number 4, 1 2 3 4, we cut at number 4, it will remain 6 that we are cutting away. Join it to that vertical 14. Are you getting now? The horizontal 14 rather, to form 20 glucose units, joined by alpha 1 4. So we have formed limit dextrin already, and the transfer of that other one to the other side is called glycan transferase. So glycan transferase will also continues by also, now the next one that glycan transferase will do is to remove, it will not count 3 again. So the process of transferring all these things is what we call glycan transferase. So glycan transferase will continue removing them very, very well. Glycan transferase will remove another one again. So we have 4 on the vertical, 20 on the horizontal. Glycan transferase will not do this again. He will not cut 3, but he will not count from the first. He will count from the down, the 4th, the 3rd, the 2nd cut. You get it now? You count. So in the first one, I can count from 10 9 8 7 until I get to the 6th one I cut. So it will remain 4 that will be hanging. But in this one now, I'll have to not count from the 4th one because it's already 4 that is hanging. I'll cut from the 2nd one because I want to leave only 1 hanging. I get it now? I'm leaving only 1 hanging. The first one is I left 4. Now I will not count from the 4th, 3rd, 2nd cuts and add it again. So glycan transferase will induce the transfer of the glucose at 1 carbon 1 alpha 1 6 and add it to what? Alpha 1 4. That's what glycan transferase will continue doing. So now when glycan transferase transfers it now the last person now is now remaining 1 hanging. So what will now happen with 1 hanging? Now the debranching enzyme, don't forget it. In the first one we have what we call branching enzyme, amylo 1416 glycosidase. Now it remains 1 that is attached by alpha 1 6 now. 1 that is attached because we have transferred 6 first one. Now we are transferred 3. It is remaining 1. So now the branching enzyme called alpha 1 6 glycosidase. We now pop open that last one by hydrolyzing it, pop it open and add it to the non-reducing end to to form glucose-1-phosphate. Remember, from glucose-1-phosphate I form UDP glucose. From UDP glucose I started donating glucose to the non-reducing end. But for this one, remember, I cut 6, add it, I cut 3, add it, it remain last one. I use alpha 1,6 glucosidase, hydrolyzes it to remove that last one to add it to the non-reducing end, forming what? Glucose-1-phosphate. You are seeing it in the picture now already. Remember, I use the debranching enzyme called alpha 1,6 glycosidase to hydrolyze the branching point. So when I hydrolyze the branching point, what am I doing now? I'm converting it to form glucose-1-phosphate. So simple. Yeah, so simple. So you see, first of all, I cut 6 out, I form a limit dextrin. I get him now. So a limit dextrin, then I now use a glycan transfer. It can transfer those 6 to the non-reducing end of alpha 1,4. Then the next one I did is that I now use also glycan transfer, cut 3 out and add it to the horizontal side already at alpha 1,4,2. Then I now use alpha 1,4 glycosidase, which is a debranching enzyme, to hydrolyze their branching point, converting that one, putting it also to form glucose-1-phosphate. So when I now form glucose-1-phosphate, what will I do? I will now rearrange. Go back, if you go watch my video on glycogenesis, what I do, from glucose-6-phosphate I rearrange to form glucose-1-phosphate. But now I'm going back. From glucose-1-phosphate I will rearrange to form glucose-6-phosphate. So I remove the phosphate from 1 and put it in 6. And anytime I'm rearranging, the enzyme is what we call phosphoglucomutase. But mutase is the key enzyme. Mutase is the key enzyme that helps us to reorganize it. So after this one, what will I do next? I'll do something next called, I will now form glucose-6-phosphate. I will now go to glucose by the enzyme glucose-6-phosphatase. Phosphatase means removing phosphate. Removing phosphate but not adding it to ADP to form ATP. That is a kinase duty. Kinase duty can add phosphate to ADP to form ATP, or take phosphate by breaking down ATP to ADP and giving that phosphate to the substrate. But in phosphatase will remove the phosphate without adding it to anybody. So glucose-6-phosphate is converted to glucose. Are you getting it now? It's converted to glucose, catalyzed by the enzyme glucose-6-phosphatase. So now I have glucose. So if I have glucose, what will I do next? I can now undergo what we call glycolysis. But before I do glycolysis, now I have glucose to be transported by GLUT4 to the muscle and the adipocyte for glycolysis reaction, or it transports by GLUT1 to the brain, or it transports just with different glucose transporters. So watch my video on digestion of carbohydrate. You see different glucose transporters. There are so many glucose transporters, but there are only 5 that are functional. So now we have formed it already. It's very, very important. You should know it. I get it now. Go to it 100%. Go to it 100%. So the liver is a place where you store glucose and where you break down glycogen. So you store glucose as glycogen and you break down that glycogen to form glucose. In that simple. So we have, again, we cut it at carbon 6. Let us say we have 14 that are joined by alpha 1,4. I get it now. And we have 10 that is joined by alpha 1,6. So you cut 6. This is me recapping now. We cut it 6, add it. You cut 3, add it. You hydrolyze with alpha 1,4, alpha 1,6 glycosidase. You pop it open at the branching point, add it. Now you have formed glucose-1,4. Are you getting now? Glucose-1-phosphate. From glucose-1-phosphate you form glucose-6-phosphate. From glucose-6-phosphate you form glucose by phosphatase. So I hope it all made sense to you and you have understood everything I've talked about in the word glycogenolysis now. So we have done it, we are done with it, we have finished everything and we have understood it. So let it make sense to you. Don't forget it. If it makes sense to you, share my video to everybody that needs it. And if you need the PDF and pictorial diagram of what you are seeing on your screen, just say it on the comment section and I will tag it to this video in my summary. Then you just take it and you can use it to do anything you want. But always subscribe to my YouTube channel every time. Share this link to everybody that will need it. Yeah. So I'll see you in my next video. Bye bye.